Imagine two patients walking into a clinic. They have the same symptoms and the same early signs of diabetes, but the diagnostic tool, powered by artificial intelligence, gets it right for one of them and wrong for the other. The difference isn't biology, it's data. Across North America, the datasets used to train diagnostic models for diabetes diagnosis are not representative of the populations they're meant to serve. Some racial groups appear thousands of times in the data, others appear only a handful of times. And when models learn mostly from one group, they become really good at diagnosing specific communities while being much less accurate for everyone else. So we asked a simple but urgent question. How does racial data imbalance affect the accuracy of diagnostic tools used for diabetes diagnosis, and what can we do about it? So to answer this, we analyzed real-world diabetes datasets and measured how well a common machine learning model performed across different racial groups. Then we built a synthetic data generation pipeline. The results were striking. We analyzed 286 diabetic and non-diabetic donors from the Human Pancreas Analysis Program and the PANCake-based program, which included 115 Caucasian subjects, 37 African American subjects, 46 Hispanic subjects, and other smaller minority groups. Already, there is a shocking disparity in participation. But before rushing into fixing this imbalance, we started by establishing our baseline. We trained a machine learning model called XGBoost to classify patients as either having type 1 diabetes, type 2 diabetes, or no diabetes, using clinical information of patients with complete records. Then we made our first attempt at fixing the imbalance by training a synthesizer called CGAN and generating entirely synthetic subjects. But this attempt actually resulted in a weaker model. Hypothesizing that CGAN's powerful modeling capacity was unsuited to the very limited samples we had available, we switched to a simpler synthesizer using real patients but with incomplete records. Our first challenge was to use a synthetic data synthesizer to fill in these blanks, while also maintaining the underlying statistical distribution. After augmenting this dataset, classifier performance improved from 88% to 98%. We then wondered if we could artificially balance the distribution by generating underrepresented patients with type 2 diabetes and type 1 diabetes. Similar improvements were found, but not statistically different because it was trained on a small data set, limiting the range of subjects to learn from and in the diversity of data points that it can generate. Our second challenge was to use the synthesizer to incorporate single-cell RNA sequencing data, which provides information on the underlying cellular and molecular drivers of the disease, including ethnicity-specific genetic variations. However, many of the donors we analyzed did not have a complete set of both clinical and single-cell RNA-seq data. We were able to bridge this gap by using synthetic data, improving our classifier performance to 94%. However, despite the promising results we obtained, the quality of the synthetic data is ultimately still limited by the data that is available. For one, limited data makes it difficult to evaluate the performance of existing models. So, when there are only a few subjects of a certain race in the test set, is the model performing well or is it purely luck? Secondly, the synthesizer learns the statistical properties of the original data. Therefore, a synthesizer trained on a dataset that is only representative of a subset of the target population can only generate data that is taken from that same subpopulation. It cannot extrapolate but beyond what has already been shown. Therefore, while synthetic data can be part of the solution, we still believe that equity must start with the reasons why there is less data for certain ethnic groups in the first place. This can be due to inequities in research participation or access, historical reasons for medical mistrust, and socioeconomic inequities. Diabetes is a condition where early detection changes lives. If diagnostic tools are less accurate for racialized communities, then the very technologies that are meant to improve healthcare actually risk reproducing or worsening existing inequities. Our work provides a framework to evaluate and correct these disparities through synthetic data generation so that future diagnostic tools are not only powerful, but fair.